All right, thank you all for coming. It is my pleasure to introduce Dr. Brian Myers from the biology program. He is a population geneticist who studies the process of speciation by looking at interconnectivity and reproductive barriers between wild populations of similar species. He began looking at these ideas while a student at San Diego State University and continued his studies as a PhD student at UC Riverside, where he helped discover a hybrid zone between Allen's hummingbirds and rufous hummingbirds. During postdoctoral studies at Cal Poly Pomona, he studied a similar effect between species of oceanic swallows. Since coming to EOU in 2023, he has returned to his work with hummingbird species, trying to determine what reproductive barriers make Allen's and rufous hummingbirds different species, and if there are aspects of human activity that promote hybridization between the two species.
Having me, my students know I really like talking about my research. So I'm going to be talking about this hybrid zone I helped discover when I was doing my PhD at San Diego State and UC Riverside, and was put on pause for a couple years, and then I came back after my postdoc, and we kind of jump-started work on the system and continued where we left off. So I study evolutionary change, and the things that drive evolutionary change range from human activity to natural processes. The environment's always changing, and species are always responding, for good or for bad. We have lots of different drivers of change, but human activity is a prominent one. We have pollution, we have agricultural land, we have the clear cutting of forests, all sorts of changes that groups have to respond to in their own way. So this has been happening ever since humans have settled, but it's really increased in the last 1,000 to 2,000 years, really rapidly. A lot of species have responded poorly, they become fragmented, migration is cut off with other populations, such as with the mountain lion, the cactus wren. Others have responded really well. If you're a crow or a house sparrow, you hang out at McDonald's and you eat French fries. So some have responded really well, others have responded poorly. So we can have different outcomes of human change, human landscape change. We can have expansions, and we can have declines, which is more likely. Adaptation to new environments and new ways of life. We can have habitat fragmentation, which can result in local extinctions of populations that have been cut off, or divergence if species are able to survive long enough to build up enough genetic diversity. One outcome I'm going to focus on is hybridization. So when we have human activity that comes in and homogenizes two different habitats, that can bring differentiated groups back into contact with each other. And it can encourage hybridization like we have with this growler bear here. If this happens, we can potentially have the collapse of species boundaries. So differentiated taxa come back into contact, and essentially one is able to outcompete the other and swamps out its genome. Before we talk about collapse of species boundaries, we have to define what a species is, and that's a source of a lot of argument in evolutionary biology. Darwin recognized how difficult this is to do, and he outlined that with this quote. So everybody knows what a species is, like you can point at it and say that's a species, but how can you differentiate that from another one? That's the tough part of having a definition that applies to these. So you can point and look and say, yeah, different species, but why? So I have a shrew and an elephant, yeah, they're different. And does that definition that you have for these two hold for these two? These are two different species, the eastern and western meadowlark. They don't interbreed at all because they don't recognize the songs that the other makes. And so, do we have a definition that can accommodate these two groups and also these two? So we have one definition, it's called the biological species concept, that establishes species on the basis of the ability to reproduce with each other. If you're reproductively isolated, so if you can't reproduce, then you're not, you're a different species. You're not the same species. If you can't interbreed, then you're the same species under this definition. Nope. All right. So this was made up in the 1960s by Ernst Mayr. Back then, they didn't really acknowledge or know how rampant hybridization was in the animal kingdom, but it is a lot more than previously thought. And so there are a lot of problems with this definition, so it's been updated recently. So as divergence happens, as species become different from each other over time, reproductive barriers accumulate, so these barriers to mating. Early on, we might have a few barriers. Later on, we have more barriers that can completely stop interbreeding from occurring. So this definition has been relaxed a little bit to accommodate what we found in recent years. And so as long as some reproductive barriers are maintained in the face of hybridization, we can still call two groups differentiated species under this definition. So now that we have this definition, let's look at what happens when we have what's called secondary contact. We have an initial set of populations that are all interconnected. Something happens and one of these populations splits off and stops exchanging genetic material with others. It differentiates over time, and something happens at some point, for example, the recession of ice sheets, that might bring diverged taxa back into contact. This is called secondary contact. And this is where reproductive barriers get tested. So we can end up with what's called reinforcement, really strong selection against hybrids. They don't do well at all, and barriers between species that are hybridizing become stronger. We can have fusion, so these species come back together, and they essentially coalesce back into a single lineage because their barriers weren't strong enough. We can also have stability, where hybrids don't perform too well, but parental species keep on coming in and interbreeding nonetheless, and this stable over time. So hybrid zones are really great to use as natural laboratories to look at how speciation happens. There's no lab manipulation required, nothing to replicate, because we're out there studying species in their own habitat. So a hybrid zone, it's a place where different ranges, or different taxa with different ranges, overlap. For example, with our frogs here, the yellow-bellied toad and the fire-bellied toad have their distinct breeding ranges, but they overlap in that narrow stretch there, and they form a hybrid zone. We can use these hybrid zones to study change over time, to study how strong reproductive barriers are, the speciation process, and we can look at characteristics that affect interactions between species. So one example of a collapse of species boundaries is with the Santa Catalina mahogany. So these species are usually at risk of collapse when you have a really small range endemic, like you do here. It's restricted to like 1 or 2 of the Channel Islands in California, and it hybridizes with the mountain mahogany, which has a really widespread distribution. So what's happening is these two interbreed on the island. The mountain mahogany totally outnumbers the Catalina island mahogany, and hybrids that they produce backcross with the mountain mahogany and not the Santa Catalina mahogany. And so over time, we're losing individuals with every subsequent generation. So this is an outcome that can definitely happen as a result of hybridization. So hybridization is rampant between hummingbirds in general and between Allen's and rufous hummingbird. Migratory Allen's hummingbird, there are 2 subspecies of Allen's. One's migratory, one's non-migratory. I'm only going to be focusing on the migratory subspecies because that's what interbreeds with rufous. So migratory Allen's is found along the California coast, along this narrow strip. In dark green, it starts here and around Ventura County, Southern California, all the way to the California-Oregon border. Rufous hummingbird takes over from there, and it breeds throughout the Pacific Northwest, as far north as Canada, Alaska. And so they overlap in red, where you see this red circle on the map. That's where they overlap and form a hybrid zone. So one of the focuses of my talk today that I'm going to be talking about more in the second half is that when we find disturbed habitat, we find a very high density of hybrids. What I look for is clear-cut areas, because I know I'm going to find birds there when I'm trying to find birds to sample. And what happens is, so parent species are adapted to different ecological niches. So when we have kind of the disruption of that and the homogenization of habitat, what we get is often a reduced selection pressure on hybrids. Because parents don't do as well in this habitat, because we have less pressure on hybrids, and sometimes hybrids perform better than either parental species because they have this novel combination of alleles. We see this in a different system between the western gull and the glaucous-winged gull, where hybrids have far higher reproductive output than either parent species. So given that hybridization is widespread, the hybrid zone spans about 300 kilometers along the coast. This has led to a lot of confusion as far as the taxonomy of these species. So the current taxonomy, I had a recent paper that agreed with this. We have migratory and non-migratory Allen's hummingbird, two subspecies, they're sister taxa. We have rufous hummingbird is next closely related to Allen's, and then Calliope hummingbird, that you see right here, is their outgroup. So this is rufus, this is Calliope. These are our two subspecies of Allen's. I can't even tell you which is which, it's so hard to tell. They vary a little bit based on size, and one's migratory and one's not. So one study in 2014 actually found that Calliope was the outgroup to Allen's hummingbird. Another study 3 years later found that Allen's and rufus was a single lineage. Shortly after that, there was a little bit of a puzzling result that came to the conclusion that migratory Allen's hummingbird is a hybrid taxon between rufous hummingbird and non-migratory Allen's hummingbird. So you can see how hybridization is really throwing things off in this system. So I believe, and something we're looking at in our research that I'm going to talk about later, that human activity is driving up rates of hybridization by largely homogenizing habitat where this hybrid zone is, and it's kind of perpetuating levels of hybridization between the two species. So if we look at Sunset Bay State Park, the landscape just near it, you don't see a whole lot of open habitat. You see a little bit. But if we go into the state park, what do you see? Hummingbirds love habitat fragmentation. More flowers, more sunlight. They don't like dense forest. They can't display in that. There aren't as many flowers to feed on in dense forests, as many bugs to eat. So when we see a campground and we have lots of open area, lots of great Himalayan blackberry that they love to feed on, and even native stuff like twinberry, we get high population densities. So this is what a campsite looks like, and that is a great hummingbird territory. You can essentially have a territory for every 1 to 2 campsites. When we have these fragmented habitats, we have lots of available space for hummingbirds to choose from. So what I'm going to address here, I'm going to address a couple questions. First, we're going to dive into some of the genetic data that we've been looking at. So given the confusing taxonomy and the widespread rates of hybridization between the two species, I want to see whether Allen's and rufus are really a single taxon. Should they be considered a single taxon, a single species, or do we have regions of the genome that are associated with reproductive isolation to kind of fit the relaxed version of the biological species concept? That's the first question I'm going to address. Then I want to see what aspects of human activity actually influence hybrid populations, and that'll I'll talk about in the second half of my talk. So as far as the genetic data goes, we have 340 samples of individuals across the range of Allen's and rufous hummingbird, as well as, of course, where they overlap. So from here to up here, to the Washington-Oregon border. We did whole genome sequencing, so we have lots and lots of data. We're working with about 1.7 genetic markers that we have. So let's address this first question. So first, before we get into this, we need to see how populations vary from each other across geographic space. So first I'm going to look at gene flow and the extent of gene flow across the system. So how often do nearby populations exchange genetic material with each other? That's essentially what gene flow is. And then we're going to look at population structure. So we want to know the ancestry of individuals within and beyond the hybrid zone. So first, let's look at gene flow. So these are some estimates I made in a program called Lamarck. These arrows you see here, if they're the same size in both directions, that means migration occurs relatively evenly in both directions. So we have just as much genetic exchange between non-migratory Allens as we do migratory Allens, so it's symmetric. If you see this big arrow here, and that's what we want to focus on, that means we have asymmetric gene flow. So it's occurring more in one direction than in the other. So what we see here is gene flow from rufous hummingbird into the hybrid zone, but not movement of genetic material from the hybrid zone into the range of rufous hummingbird. So there's something acting to stop that. But what we do see is that asymmetric pattern from rufous to the hybrid zone, but then a symmetric pattern between the hybrid zone and migratory allens. So what that means is we have alleles going from here to here to here. And so now let's talk a little bit about population structure. So I want to know the genetic ancestry of individuals within and beyond the hybrid zone, and that's what this analysis is going to do. So on these plots here, each individual bar that you see designates a different individual in the data set. We have genetic ancestry on the y-axis right here, and each color corresponds a different genetic cluster that an individual belongs to. So we have 3 distinct clusters here. So what's that? We have 100% peach ancestry here, but we have, with these individuals, 50% red from cluster 2 and 50% peach from cluster 3. So these are probably hybrids. They have mixed ancestry. So here's the actual data. What we have here, on the x-axis, we have counties from north on the right to south. We have our genetic ancestry on the y-axis. And so this is the range of rufous hummingbird up here. So we don't see much mixed ancestry outside of the phenotypic hybrid zone throughout the range of rufous. And this phenotypic hybrid zone, that's based on morphological and behavioral traits. So that's kind of our baseline that we're working with here. So we don't see any movement from the hybrid zone, from the phenotypic hybrid zone into the range of rufous, 100% ancestry. We see lots of evidence of admixture within the hybrid zone. But the really interesting thing is when we get south of that hybrid zone, whose alleles do we still have all over the place? As far south as Monterey, California. So when you look at this map, that's crazy. This is the hybrid zone here. Rufous alleles find their way all the way there. We only have one parental migratory Allen's hummingbird population that doesn't have admixture with rufous hummingbird. And that's in San Luis Obispo, which we're sampling next week. And we're going to find out when we sequence those individuals if they have rufous alleles now, 10 years later. So as far as the first question that I'm talking about, that seems a little troublesome on its face. Is fusion possible? So far, kind of looks like it might be possible. But when we run this same analysis using only the Z chromosome, so a sex chromosome in birds, we get a totally different pattern. So we have a similar pattern with rufous here. Some evidence of admixture in the hybrid zone. Very little evidence of admixture outside of the hybrid zone into the range of Allens. So what it looks like is the Z chromosome has some sort of selection acting on it that's preventing alleles on the Z from rufous from pouring into the range of migratory Allens. So that kind of puts us in a direction to do some other analyses. So first, why do we think this pattern is driven on the Z? So in birds, a lot of reproductively important, reproductively isolating loci and traits typically trace back to the Z chromosome. In bird, sexual selection is an incredibly important driver of speciation. And it's what drives a lot of differences between birds. We see a lot of differences in plumage coloration, and courtship displays, things like that, things to attract mates. So as Darwin defined it, we have natural selection, which is your ability to pass on, survive long enough to pass on your beneficial traits to your offspring. Sexual selection is only concerned with attracting mates. And typically, traits undergoing sexual selection trace back to the Z in birds. So starting from that point and jumping off from there, we're going to investigate whether traits on the Z are under reproductive isolation or not. So first, I'm going to use these ancestry estimates from admixture for future analysis that I'll talk about in a second. Then I'm going to look at what are called fixed differences between Allen's and rufous hummingbird. So say we have, in a really simple example, say we have a gene for eye color. We have an allele, a version of the gene, that codes for blue eye color, and another that codes for brown eye color. If we have one species that has only blue eye alleles and another that only has brown eye alleles, they're fixed. They have a fixed difference at that locus or location of the genome. So these are traits that could be due to selection, but also maybe not. So it would need to be tested. But there are definitely differences between species. And this is what we see with migratory allens and rufous. So we have 322 fixed differences. Look at where most of them are. How suspicious. They're mostly on the Z chromosome. Okay? So we know that. So now I'm going to talk about what's called geographic Klein analysis, and this is kind of the heart of a lot of the talk today. So what this does is this is analysis that tells us about reproductive barriers between species. So these are measure of variation across geographic space. So every point you see here is an average value of a population for a trait. So if you see this fairly wide slope, it looks kind of like a slide. That's a wide Klein. So that means we have a lot of hybridization happening. So we have a parental trait that we're looking at on the y-axis, the trait value. So this is an allele frequency. So this is a fixed difference, right? In this species, that frequency is at 100%. In this species, it's at 0%. So they're fixed for different alleles in their parental ranges. Where they hybridize, we see mixed ancestry, right? We have about 70% ancestry for that allele in that population. And so we see evidence of hybridization spanning roughly 180 kilometers to 340 kilometers. So almost 200 kilometers, we see hybridization happening. So that's what that means. So if we see a relatively narrow Klein, so you see that slope got a lot steeper, that means hybridization is restricted to a smaller geographic area. Because we're right around 250 and we don't move far beyond there. So if we look at traits and we see very few intermediate traits, for example, plumage coloration, what that means is this species does not like the plumage coloration of this species and vice versa. That trait is not passing through this hybrid zone because this species does not want to mate with hybrids that look like this one. But if that's the case, we kind of have a free-for-all. So we have interbreeding between parental taxa and hybrids, and it doesn't seem that they discriminate all that much. What happens when we get what's called a coincident Klein between a genetic locus and a trait? So when they're Klein center, so the Klein center is the steepest part of this graph here. So right there about is the Klein center. When we have concordance of narrow Kleins, because they're responding the same, this could imply that that locus that is under reproductive isolation could be associated with the phenotypic differences observed between species. And that's kind of the key. So I'm going to be using these admixture results. Remember, each bar is an individual. And what I can do is I can use the ancestry of each individual to take an average value for each population and make a Klein based on their ancestry. So I did that for the whole genome. So the whole genome admixture plot ancestry, I did it for the Z chromosome, for just the non-sex, for the autosome, so the non-sex chromosomes, and we'll see what they look like. So the whole genome, it doesn't look that wide, but that's pretty wide, 500 kilometers almost in width. So pretty relaxed amount of selection, it seems like, is our baseline that we're running with. And this makes sense given what we saw with gene flow, given what we saw with our admixture plots. The autosomes are even wider. So this is everything excluding the sex chromosome, the Z, that we're focused on. So that is incredibly wide. It never levels off. And that's similar with what we see in our parent taxa. We have lots of rufous alleles way south in the range of Allens. Look what happens when we look at the Z chromosome. It's way narrower. So that drops to about 120 kilometers, 119 kilometers. So we don't see individuals who are hybrid going beyond this narrow strip. And if we look at an individual locus on the Z chromosome, based on fixed differences, so remember how I calculated those fixed differences, we are then running geographic lines on allele frequencies of those differences between parent species. So what we found for this one, this is for a coding region on a gene called GAK. This cline is very similar to that of the Z chromosome. So something interesting is going on the Z. It looks like the Z chromosome definitely houses loci that are important to reproductive isolation between the species. So we have some sort of barriers that are acting. So these clines, reproductive isolating when they're really steep, far along in the speciation process, relative to clines for characters that are really relaxed and have a wide slope. So now we know what these loci are. We have a really long list of them. What are they and what do they do? That's the fun part. So 2 undergrads here at EOU, one of them in the room, Preston, have been working on this in research. And we've found some really interesting things. So we have these fixed differences between the species. We test whether they are actually contributing to reproductive isolation by running those clines. And so when we have a locus that corresponds to a really steep cline, Preston and Tyler then go in and they find out what that locus does, and they do a big literature search and try to determine why it might be under sexual selection. But it's really difficult to pinpoint the exact reason. We can only come up with hypotheses. So what are these loci and what do they do? So this is a lot of the information that we have to start with. It's not incredibly helpful for this purpose. We have the chromosome, we have the location of each, like the relative location of bases on the chromosome, we have genotype information. So this is unannotated. So that means there's no gene information. We don't know whether we have an exon or a coding region, an intron, an intergenic region. We don't know a whole lot at all. So what we do is we take what's called an annotated reference genome. In this case, it's from the Anna's hummingbird that has all of this information. And because our species are pretty closely related with Anna's, we can use that and overlay that on our data. And this is what we end up with, which is way more helpful. So we end up with the relative location of each locus, what it corresponds with. So this intron, which is part of a gene, but it doesn't directly code for a protein, but it's a part of DMRT3. So we know that, and so if we want to focus on DMRT3, we can go onto the NCBI database. We can look up our Anna's hummingbird reference genome, locate it, and you just click on that, and we get some information about what it does. Even more helpful is doing a literature search and finding out what other authors have found. What does this gene do? What's it involved with in different taxa? So we just use this. It's not an end-all answer, of course. Lots of testing needs to be done, but it builds a hypothesis. So GAK, one locus with a really steep cline, it's involved in behavioral response. And that's a pretty interesting result that I'll cover why in a couple slides. We also have an intron as part of TRABD2A. So this inhibits what's called the WNT signaling pathway. And in birds, this pathway regulates feather development and maintenance, specifically barbial, hooklet, and rachis development. So you can notice that those two species are pretty different colors. One's orange, one has a lot of green on it. So we think that's sexually selected, and this is an interesting result. We have another one, CPLX1. This codes for complexin. So this has been found to affect social behavior in mice. So all three that I'm going to cover right now, they're involved in feather morphology and behavioral responses. One interesting thing is all of these loci have the same Klein center. They line up in the same place. And so what I'm going to do next is I'm going to show you some clines of phenotypic characters that are based on courtship displays. So we have a couple of courtship behaviors that I have clines for here. One is dive sound frequency. So the males typically fly up really high, we fly down, and they make this tail feather sound. So dive sound frequency is the sound that they make when they do that dive. And it's different between both species. There's also another display that's a little bit harder to explain. It's called they make this weird clicking sound after they're done doing the dive, and that has a different number between each parent species as well. And then I have another cline right here that's based on bat color. Look at their cline centers, 964 kilometers. So they are pretty concordant with what we see with our genetic loci, with our loci. And these can all be placed in Coos County along the coast, right where Sunset Bay State Park is. So when we sample in these places, the pretty striking thing we noticed in the field is when we go from Sunset Bay to Bullards Beach State Park, it's a span of about 10 kilometers, we go from hybrids that look and act like rufous to hybrids that look and act like Allen's hummingbird. So it's an interesting place for this cline center to be. So phenotypes, of course, it's not as easy as what you might have learned when you learned Mendelian genetics a long time ago. We have different molecular interactions that correspond to a phenotype, right? It's not this one gene causes this phenotype. It would be much easier and much less interesting if that was the case. So it's unlikely that any individual region directly corresponds or solely drives a given phenotype, but we have some interesting associations here. So we have these three loci that are lining up with those three phenotypic traits that I outlined. And there are more loci that are at that cline center, and there are more phenotypic traits based on courtship behaviors that also line up at that center. So now we're trying to disentangle all of that. But it looks like these loci are involved in reproductive isolation, as are the traits, and could, with an asterisk, be associated with the observed phenotypic traits that we're seeing. But we need to do more. Interestingly, kind of a side note, collaborators at UC Riverside are doing what's called a GWAS study on this. So that's a genome-wide association study. So they're looking for associations between underlying genetic traits and the physical traits that we observe. And they have correspondence to those behaviors I talked about with these three loci. So it's a good start. So Darwin knew that it was difficult to define what a species is, right? And we still agree with that to this day, because you can get any evolutionary biologist to argue about what a species is. Meyer had a pretty good, pretty durable definition that we've adjusted. And so because we do see these reproductive barriers between Allens and rufous, we think they're young species. We have estimates of divergence dates that range from 80,000 generations ago to 140 or so thousand generations ago, which in evolutionary time is very young. But they have reproductive barriers, areas of the genome under reproductive isolation that are not at all being swayed by the hybrid zone. So we have good, although young species. So now let's pivot to the next question. So what aspects of human activity influence populations? Not as much data for this one. We've only had one field season so far, but we do have a pretty cool result. So North American hummingbirds have undergone lots and lots of range expansions associated with human activity. Non-migratory Allen's hummingbird, you're more likely to find it in your backyard than in the wild. Anna's hummingbird, its range was restricted to California in the 1940s, and now it's in Canada. It winters in La Grande because Laura and Trent have feeders out in La Grande, so they decide to stay. So human activity definitely benefits these groups. There's no arguing that. So what we're finding in the literature is that human activity tends to homogenize habitat that used to promote these niche differences between species. But altering habitat eliminates these differences a lot of the time, and so hybrids tend to perform a bit better in these areas. They have, again, more relaxed selection pressures, and in some cases, they outperform parents because they have new combinations of alleles either parent doesn't have, so if they're beneficial, they're going to do well. And we have multiple individuals with different combinations of alleles. So there's lots of evidence supporting the fact that migratory allens and rufous both benefit from human activity. If you want to find them, just go find a clear-cut forest or go to someone's backyard that has a feeder, and you're likely to find them. Their hybrids are very common in disturbed habitat. In the hybrid zone, along the Oregon coast, the easiest place to find hybrids is at state parks, where we have habitat fragmentation because we have campgrounds and day use areas and all of these nice flowers that are able to take over because we don't have this dense forest canopy anymore. So if I pass a state park, I know I'm going to find a bird there. In between the state parks, they're much more difficult to find, where land is less disturbed. So I think human activity might increase hybrid success. So just a quick recap. We have alleles from rufous present across most of the range of migratory allens. Genetic differentiation across the entire system is pretty low. Like they share a lot in common across the genome. We showed really high gene flow across the system, especially from rufous into allens. And humans definitely influence the species, and they might influence the hybrid zone. So to address this question, still in progress, one, we want to see whether population density of hummingbirds is influenced by human activity. That's question number one. And then question number two is we want to see whether human activity influences diet. So to start with question one, last spring we sampled 9 localities throughout Oregon, mostly along the Oregon coast. So that's Dylan and Cameron, 2 students who've been doing a lot of work on this project. So we sampled a perennial rufous hummingbird, a couple of those populations at the Oregon-Washington border in orange on the map, and then hybrid populations from central to southern Oregon. We were busy. So Dylan's job was to perform line transect sampling at each locality. So we set up 5 transect lines at a location and ran 5 points per line. He made observations at each point on the transect line for about 15 minutes, and he recorded males on a territory, on a breeding territory that they vigorously defend, or whether there was a non-territorial male flying around or visiting the territory or something like that. We don't bother trying to count the females because they're very difficult to detect. Males want to be seen, females typically don't want to be seen, they just try to sneak a meal without getting bothered. So what we do is we estimate total population density by assuming a 1 to 1.5 male to female ratio. And so for each transect line, we total the total number of observed males, and that's our metric of population density. For human activity, we're starting out by testing for associations between hummingbird population densities we calculate and human land use in the area. So what we focused on are permanent residents, so who lives in the area permanently, there's a lot of houses, apartments, whatever. The number of campers per year, which tells us how large a campground is and how heavily used it is, and the level of day use within a sampling location. And Dylan got all of this information directly from Oregon State Parks, as well as from census data for the number of residents. What Cameron focused on was the vegetation in each territory and the frequency of visitation by hummingbirds who weren't on the territory, who weren't defending it. Because what will happen is a male will defend a territory, he wants to guard the resources, he doesn't want anyone else to use those resources. So what happens is invaders come in, they try to sneak meals, and it's the male's job on the territory to chase them off. So he looked at frequency of visitation by non-territorial hummingbirds to a male's territory, and delineated what a territory was, or what one territory started and ended versus when another started and ended, by looking at the area that a male would defend, and he evaluated that for about an hour. Then he quantified the area and perimeter of each territory using a rangefinder. So we classified vegetation within a territory as native or invasive or non-native. When possible, we identified plants to species level and also recorded the number and sex of hummingbird visitors as well as the resource the invader tried to utilize. So we're trying to build data to, one, see how often invasive plants are found in a heavily used territory versus native plants, and also we're trying to get information on habitat quality or territory quality, but that's not going to be a full data set for a long time. So we used what are called generalized linear models to look for associations for Dillon's data set between hummingbird population densities and the number of day-use visitors, so our human activity metrics, the number of campers, and the number of permanent residents. Then we did a similar analysis for Cameron's data and looked for association between visitation and aspects of the territory. So native vegetation area, the area of non-native vegetation, territory perimeter, percent of non-native vegetation, and percent native vegetation. We do have one result, very little data, so this is still exciting. We have a significantly positive association between campground usage and hummingbird population density. This also does make the most sense to me because remember when I showed that image of Sunset Bay State Park, what do you see? Lots of fragmentation, lots of cutoff areas with lots of flowers, lots of open space relative to the rest of the forest that is relatively undisturbed and just has a dense canopy. And the place to find a hummingbird when you are not in a state park is usually along a creek, along a river, in a meadow, and of course this is observational. We're getting data from non-disturbed areas with this next field trip. Density seems to be a lot lower, but okay, again, I haven't tested that yet. We don't have any significant associations between permanent residency or day use. But again, we have a pretty small data set so far. And unfortunately, we found nothing between characteristics of the habitat and hummingbird visitation. That's okay. Again, we have a very limited data set. But we do have that one really cool result. So the next question that we're still in data collection for is, does human activity influence diet? So feeders are really common, especially in developed areas, and they provide a large non-protein portion of a hummingbird's diet. They eat bugs as well for protein, but when feeders are available, they offer a pretty good caloric value to them. So most feeders are filled with a sucrose solution that's from sugarcane, and so it has a distinct carbon stable isotope ratio from what's found in flowers. And so by taking blood samples of individuals across the hybrid zone and in the parental ranges, we're going to look for associations between higher feeder use and more developed areas and population densities. So far, we have 21 samples that were acquired last year. I'm hoping to get I'm going to double my total by the end of this field season. We're going to continue sampling. And so I want to compare this coastal transect, which is incredibly populated relative to going inland along the Klamath River. So this is relatively undisturbed. I want to look at diet there, and I want to compare it to diet along the coastal transect, which has a lot higher human activity and human population density. I also want to see if it's just a thing with hybrids, or do we see the same pattern in parents as well? We probably do. So I want to sample lots of rufous hummingbird populations and lots of Allen's hummingbird populations far from the hybrid zone, and see if we get associations between density and feeder use. So for our first question, swamping seems pretty unlikely because we have these areas mostly on the Z chromosome, but we do have fixed differences elsewhere. That suggests these loci have a role in reproductive isolation between the two species. So they are able to withstand incredibly large amounts of hybridization. So these are good species under our biological species concept. For question number 2, we did find this one cool result, but more still to come, maybe next year. And so next steps in this study, for the Klein part of things, you never trust a single Klein because that's just one set of data. So we ran this all for the coastal transect. We have another inland transect we have data from. That's along this Klamath River, right here. So we need to do data analysis of that data that we have. And what we want to see is if we see the same concordance between our genetic loci and our phenotypic characters on that inland cline that we see on the coastal cline. So do we see the same result independent of environment? I also want to back that up by performing explicit tests for selection across the genome. The tests I plan to do are called haplotype scans. So these are tests that we'll do across the whole genome, and we'll see if these same genetic regions pop out just like they do in our cline analysis. And as I mentioned earlier, colleagues at UC Riverside are currently doing a GWAS study, and they are finding associations between many of the loci that I found and the phenotypic characters that I talked about today. So they're finding associations between genetic loci on the Z and the courtship behaviors we observe selection on. For the second half of the talk, I'm going to continue analyzing carbon isotope data from blood samples. And I want to test the questions that we cover on that project separately for perennial allens, perennial rufous, and our hybrids to answer the question of whether human activity perpetuates hybridization, we need to independently test parent populations as well as hybrid populations and compare them. So we want to increase sampling overall, sample lots of parent populations, and then independently evaluate the parental and hybrid populations we're looking at. So I'd like to thank all of the sources of funding starting from like 10 years ago to today, all of my students past and present, and thank you for coming to my talk, and I'd be happy to take any questions.
Speaker.
So pretty cool, Brian. I have a question about the hybridization zone, and you're getting a very steep climb on Z and sexual characteristics, but you're also getting expansion of the rufous phenotypes and genotypes going further south.
Yeah.
But you're not seeing the non-migratory characteristics going north, right?
Right.
And so what I'm wondering is, if, say, the females rufous or the female hybrids only prefer rufous hummingbirds, whether that could create a very steep sexual climb, but allow for expansion going south as the maybe the hybrids are also have some aspect of migration going south as well. And what you're going to see is that steep climb moving south over time.
That's a good question. It's something we thought about. That's also why I still want to be collecting data, because I want to see if the hybrid zone is moving. So one thing you do is you try to replicate in some places where you've sampled before to see if that center's moved. And one future step we want to take is looking at museum specimens and old tissue collections to see where this center might have been before. So that's one thing we're looking into. As far as, so it looks like a rufous hummingbird doesn't like anything hybrid. So that's why we don't see those alleles passing from the hybrid zone into the rufous side of things. On the flip side, what the data is saying is that Allen's hummingbird doesn't like hybrids that behave like rufous, but if we can have hybrids that don't behave like rufus, they're free to interbreed with them, and that's kind of what we see happening. So that's why we see those courtship traits trapped in the hybrid zone, but everything else is kind of just moving through. So it looks like they're really important barriers.
I guess two things, whatever. You probably already established this. Is it possible that the females just aren't as mobile on the rufous, or did you find plenty of rufous females in the hybrid zone?
Yeah, we find plenty of them. They're just very cryptic. So usually when I see them, they're sneaking a meal at the feeder right before dark, right before bedtime, and they're disappearing. So we see them. I can't quantify how many there are. They're very difficult to deal with, but they're present.
And the other thing was just, do you have some hypotheses about human activity? I mean, these parks are going to be gardened and cleared out and open, but it seems like you're seeing if there's more people there, there's still more birds on account of that, seemingly.
I don't think it has to do with the number of people actually camping. I just think it has to do with having more campsites at a campground that has more people. So if we have higher visitation at a campground, usually all of these Oregon State parks, they book far in advance. So they're usually all full. So if we have higher visitation in one park than another, what that probably means is the campground is larger. So we just have more of this fragmented habitat. So more ideal habitat if you're a hummingbird. Did that answer your question?
Other questions?
So I got a question about the weather. And weather at the coast. Maybe it's always rainy. But does the weather impact when you're testing for numbers of numbers of birds? Does it, if it's a rainy day versus a sunny day, do you have to allow for that, or doesn't it matter?
The convenient thing for me and the inconvenient thing for them is if they don't eat, they starve to death fast. So they have to go to the feeder at some point if it's a rainy day. They typically display unless it's downpouring. Dylan learned this the hard way when we're out there with our hoods on looking sad, waiting for a hummingbird to go to the feeder. So they care, but not as much as you might think. So they're still pretty active.
All right, thanks.
Yep.
Can you share a little bit more about how you decided to look at the Klamath River drainage also, and what kind of work you've done already up there, and just like anecdotally or observationally, what differences you're seeing?
Yeah, so we've mostly covered the coast, but we've completed sampling with the Klamath River, and there are some pretty interesting things there. We chose it because it's the only inland area we could find where we didn't have an really abrupt elevational gradient, because Allen's hummingbird won't breed above 500 meters in elevation, or a little higher than that, whereas rufus is more flexible, so it can be found from low to high elevations. So we didn't want to disturb the cline by sampling along a gradient where we're going up and down and up and down. So if we follow the Klamath, we can kind of stick to this low elevation sampling scheme. There's this, are you familiar with the Klamath area? Okay, so there's this place called Happy Camp, and that's actually a really interesting spot because it's the only locality where I sampled a perennial rufus, a perennial Allen's, and a hybrid in the same place. So that was really cool. I was excited about that. Another thing that happens that's kind of interesting is we get Allen's, then we get hybrids, but it's hard to find that perennial rufus population. It just turns into Anna's hummingbird habitat. And so to make up for that, I had to go all the way to Josephine County to complete the transect line, because that's when we find rufus all over again. And so with all of this interesting stuff going on, it'll be really interesting to see whether that genetic data still corresponds to the phenotypic data in the same way. Yeah.
So you're sampling using hall traps, right?
Yeah.
Okay. So I know from some of my sabbatical work, do you remove all feeders and only have one feeder with your hall trap in it, or does everybody still have all their feeders up when you're trying to do your sampling?
It depends. You can tell if a population is going to be annoying pretty quick. Some populations, if they know there are other feeders around, they're not going to go into this big scary thing. And so they're going to go to those feeders. And so I have to take all the feeders down. Sometimes they'll then go to the feeders on the floor and I have to literally put them away. Then they kind of reluctantly go to the trap. Other populations, they just don't care. They're just going to go. And right after I sample them, sometimes I find them right back at the feeder again. So it's clearly not that traumatizing.
Maybe time for one more question.
So if I heard you right, you're going on a field trip soon, next week, week after, something like that. And you said something about sampling in an undisturbed site, and you also suggested that might be on the Central Coast?
No. So
Okay.
we're going to start on the Central Coast because I need parental allens, migratory allens, and that's going to be in San Luis Obispo and Pismo. But then we're going to dart to the Klamath region.
Oh, okay.
Yeah, so very low population densities. In some areas, it's zero, like 0 to 20 humans. And we have to sample along the river because that's the only place you're going to find the birds. So it's much more natural than
Okay.
yeah, the coast.
Thank you.
One more, maybe?
I'm just curious about. I mean, the Anna's is bigger and more aggressive. Is that fair? Maybe not. But
They
the-
all have their, yeah, go ahead.
oh, the question is, you know, how do Annas factor into this dynamic? Yeah, how does behavior change where Annas are more dominant or present,
Yeah,
or are they
so
everywhere?
there are stereotypes about the three species. Rufus is allegedly the meanest, but sometimes Anna's is the meanest. It's really variable depending on the individual at the end of the day. The biggest interaction that I see is when an Anna's will guard the feeder and not let Rufus go to it. So I sometimes put Anna's in timeout, where I'll put it in the banding bag for a minute, and then I'll wait till I can catch the Rufus. Then I'll give the Anna's a meal and let it go. That's usually what I see. I know Anna's hybridizes with Allens sometimes. It's not widespread. It's kind of more of a one-off thing. I don't know about with Rufus. I wouldn't doubt it. But yeah, that's pretty much the extent of it. They're sometimes found in the same habitat with Allens, not as often with Rufus.
I think that is all the time we have, so let's thank our speaker again.